Welcome to the Explainer. We're going to clear up two key lab terms, validation and verification. So, are they the same thing? Not at all. And getting them right is absolutely critical for patient safety. Okay, so here's the breakdown. If a lab basically builds its own test from the ground up, that's what we call a laboratory developed test, or LDT, or if they take an existing FDA-approved test and tweak it, they have to do a full-blown validation. You can think of it like this. The lab is basically the manufacturer now. They have to prove everything about that test works, starting from scratch. But if they're just using a standard, off-the-shelf test kit that's already FDA-approved, exactly like it says in the box, then their job is to verify it. They just have to prove that the manufacturer's claims are true in their lab.
And just so we're all on the same page, an LDT is a test that is born and lives its entire life inside of one single laboratory. Now, why would a lab even do this? Well, a lot of the time it's for things like rare diseases, where there just isn't a big commercial test kit available, so they're filling a really critical need. But since it hasn't gone through that big public FDA approval process, that one lab carries all the weight of proving that it's reliable and accurate.
A full validation. Wow, it is a massive undertaking. We're talking about a whole battery of different studies. The lab has to establish its accuracy, does it give the right answer, and its precision, does it give that same right answer over and over and over again. They have to figure out its reportable range. What's the lowest and highest number it can measure reliably? They even have to figure out what a normal result looks like for their specific patient population. It's a truly ground-up, exhaustive evaluation.
And it doesn't even stop there. Depending on the test, they might have to look into all sorts of other things, like could a common medication totally throw off the result? That's an interference study. Or they check for carryover to make sure a little bit of your sample doesn't get into the next person's. They test how long the sample is stable for. It goes on and on. They're literally writing the entire instruction manual for this test themselves.
Okay, now let's hop over to that other path, verification. So if a lab is just using a standard FDA-approved kit, the good news is the manufacturer has already done all that heavy lifting, that massive validation. The lab's job isn't to reinvent the wheel. It's to prove that the wheel they bought works perfectly on their car, in their garage. They just need to verify the claims the manufacturer already made.
See how much shorter this list is? The lab just has to confirm these 4 key claims. That's it. Oh, and a quick but really important side note about tests that get an emergency use authorization, or EUA, like a lot of the tests we saw during the pandemic. These are not considered LDTs, so they only need verification. Now, during the actual emergency, that verification might be a little more limited if supplies are tight. But here's the key. As soon as that test gets full FDA approval, the lab has to go back and do the complete, full verification process.
Okay, so far, everything we've talked about is for tests that spit out a number. But what about all those tests that just give a simple yes or no answer, a positive or a negative? Well, figuring out if those tests work presents its own unique challenge.
The standard tool for this is a simple but really powerful little grid called a 4-fold table. Basically, the lab takes a bunch of samples and runs them on both the new test and a trusted old test. Then they sort the results into 4 little boxes. Box A is where both tests said positive, box D is where both said negative, and then boxes B and C are for when the 2 tests disagreed.
And from that little table, they use this super simple formula. They just add up all the times the tests agreed, that's A plus D, and divide that by the total number of samples they ran. That gives them a percentage, which tells them how well the 2 tests correlate.
So what's the magic number they're hoping to see? Almost always, they are looking for a correlation that's greater than 95%. That's kind of the industry benchmark. If they hit that number, it gives them real confidence that their new test is performing just as well as the old one they already trust.
Of course, that's not the only way they can do it. Labs have a few other tools in their toolbox. They can use special samples where the answer is already known, we call those proficiency samples, or they can even compare the test result to a patient's actual clinical diagnosis to see if it all makes sense in the real world.
Now here's where things get kind of cool. Words that you might think you know, like sensitivity and specificity, actually have a slightly different meaning for these tests. For a yes-no test, sensitivity isn't about detecting the tiniest amount of something. It's about defining the cutoff, the exact line in the sand that separates a negative from a positive. And specificity is all about not having false positives. It's the test's ability to correctly say no when the answer really is no.
So who comes up with all these rules anyway? This whole system of validation and verification isn't just a good idea. It's the law. It's all dictated by something called the Clinical Laboratory Improvement Amendments, or CLI, and it governs every single clinical lab in the country. And making sure CLI works is actually a team effort between 3 different federal agencies. You've got the FDA, which categorizes how complex tests are and helps develop the rules. Then you have CMS, the Centers for Medicare and Medicaid Services, who actually publish the regulations and inspect the labs. And finally, you have the CDC, which provides all the scientific research and technical backup for the whole system. It's this 3-part system designed to guarantee quality.
Okay, let's kick things off with a challenge that labs face all the time. See, most tests are designed and approved for really standard stuff, like blood or urine. But what happens when a doctor needs a test run on something a little different? Exactly this. A test might work perfectly on blood, but the unique chemical soup of something like spinal fluid, scientists call this the matrix, could totally mess with the results. So the lab can't just hope for the best. They have to prove that the test still works, either by finding research someone else has already done, or by rolling up their sleeves and doing their own validation. So how in the world do they do that? Well, 2 of the most common ways are dilution studies and spiking. With spiking, a lab literally adds a super precise amount of the substance they're trying to measure, that's the analyte, into a sample. Then they see if the test can find the correct amount. The trick is to add just a tiny bit, less than 10% of the sample's volume, so you don't accidentally change the sample's chemistry. But here's the absolute bottom line for all of this. It's not just about passing some statistical test. The number 1 most important factor is clinical relevance. I mean, think about it. A test that's 99% accurate on paper but misses a crucial disease threshold is worse than useless. It's downright dangerous. That's why the lab's medical director is always involved, to make sure every single strategy is built around patient safety. And if, for some reason, a full validation just isn't possible, well, the lab can't just wing it. They are required to add this exact disclaimer to the patient's report. This is just a fantastic example of transparency, right? It's a direct message to the doctor that basically says, hey, this number might be helpful, but you've got to use your own clinical judgment to figure out what it really means. Okay, moving on. It turns out it's not just the unapproved, unusual samples that get this kind of intense scrutiny. Even your standard, fully approved everyday tests sometimes have to be completely re-verified. There are certain events that can trigger a whole system recheck. And it makes perfect sense when you think about it. Just moving a super sensitive instrument from one side of the lab to the other could change its performance because of tiny shifts in temperature or humidity. If a lab buys a second identical machine, they have to prove it gives the exact same results as the first one. Or what about bringing back a seasonal flu test that's been sitting on the shelf for months? Yep, that needs to be re-verified. And of course, adding a totally new test to a machine you already have, that requires its own full verification from top to bottom. This brings us to the person who's actually doing all this work. And it's not just about, running samples and crunching numbers. The validator has a really clear 5-step playbook they have to follow to make sure the whole process is objective and actually means something. And guess what? It all starts right back at that clinical perspective. How is this test going to be used for real patients? From there, they set a super clear performance target before they even start testing. Then they gather the right kind of data, use stats to figure out the method's error, and finally, they compare that error to the target they set and make a final objective call. It's a really beautiful logical flow. Okay, let's zoom in on that second step because it's a big one. Setting a performance target. Before a single experiment is run, before any data is collected, the lab has to define what success even looks like. What's the acceptable margin of error for this one specific test? And that brings us to this absolutely critical term, allowable total error, or TEA. This isn't some arbitrary number they pull out of a hat. It's the maximum amount of error a test can have before the results become medically misleading. You can think of it as the line in the sand between a result that's useful and one that's actually dangerous, especially around those critical levels that decide if someone is healthy or sick. So where do these targets even come from? Well, labs don't just make them up. They pull from a whole hierarchy of sources. Clinical needs are always at the top, but they also use published recommendations from professional groups, goals set by regulatory bodies like CLIA, and criteria from proficiency testing programs, which are basically like pop quizzes to make sure labs are staying accurate. All right, so we've set our target, we know what we're aiming for, now comes the moment of truth, the final calculation. This is where the lab measures the actual error of their test to see if it fits inside that allowable error target we just talked about. The number they're trying to figure out is called the total error, and it's really made of 2 parts. The first is systematic error or bias.
That's a consistent error, kind of like a scale that's always 5 pounds off no matter who steps on it. The second part is random error or imprecision, and that's the unpredictable little bit of scatter you get even when you measure the exact same thing over and over. Total error combines both of those to give you the whole picture. Now, when setting that allowable total error target, labs often look to two main places. On one side, you've got the proficiency testing limits, which are set by regulatory programs. But on the other, you have goals based on biological variation, which is just the natural ups and downs a substance has inside the human body. And here's the key. Those goals based on biological variation are often way, way stricter and more relevant to what doctors actually need. Let's look at some real-world examples of those proficiency testing limits to see what I mean. For a liver enzyme called AST, the allowable error is a percentage, plus or minus 20%. For calcium, it's a fixed number, plus or minus 1 milligram per deciliter. And for glucose, it's actually a combo of the two. This just shows how these targets are totally customized for each specific test. But here's the really important nuance. That calcium limit of plus or minus 1.0 milligrams per deciliter, for a doctor who's making a critical decision about a patient's heart, where tiny little shifts matter a whole lot, that can feel like a massive margin of error. They often need to test with way less wobble to see the changes that are truly meaningful for a patient. That's why those biological variation targets are so important. And after all the experiments, all the statistics, all the work, it all comes down to this one simple, powerful rule. The final calculated total error of the method has to be less than the allowable total error they decided on at the very beginning. If it is, the test passes. If it isn't, it fails. That's it. So what happens if it fails? Well, it's a hard stop. The lab has to figure out what went wrong, and they'll usually work with the manufacturer to troubleshoot it. Maybe they can fix the problem and try again, but if that method keeps on failing, it gets rejected. Completely. It will not be used on patients. Period.